New tools have also been developed in the core breeding domain for Milestone 6. Plus, users will be able to enjoy new and improved features in existing tools. One key new feature in the experiment creation tool are cross patterns. When breeding teams are making crosses with parents that are repeated several times, such as in the case of making hybrid test crosses in maize, they often want to plant the repeated parent multiple times throughout the field to support many crosses. Now users can choose from two popular cross patterns when they are making their nurseries to efficiently define their crosses and the planting arrangement of their parents in a single step. After users create their experiments in the EBS, they will be able to turn to the Planting Instructions Manager tool to support seed packing processes. This tool tracks seed quantities when seeds for planting are removed from source inventory items. And it also provides an important opportunity for users to make substitutions for germplasm when necessary in a controlled and traceable manner so that the right materials get planted and the correct germplasm information is available for analysis is in every plot. Once users initiate their experiments, they can continue to monitor and add additional data to their occurrences throughout the season using the Experiment Manager tool. Breeding teams can also now use this tool to add new occurrences to their experiments if they have the opportunity to plant in another site after they have completed their initial experimental setup. Plus, breeding teams can now share specific occurrences to registered collaborators directly within the EBS. These collaborators can then capture the data and load it directly back to the EBS to enable efficient data sharing within breeding networks. Data curation tools have also been enhanced for Milestone 6. Users can now clearly see data points that are out of range at the moment that the data are uploaded. Plus, they can directly update any errors in the collected data directly in the data terminal before the data are committed to the database. This provides a first level of quality control as data are uploaded throughout the season, but another level of pre-analysis data curation is also under development in the Phenotypic Data Manager tool. It will be released for review and feedback under Milestone 6. Manager continued to implement new use cases to support the basic harvest processes required by rice, maize, and wheat breeding programs. After teams have harvested their seed, the new Inventory Manager tool enables admin users to update information about seed quantities and their storage location. And in February, all users will be able to easily share seed between breeding teams while maintaining full traceability of their materials through an internal seed transfer process. New printing functionalities are also now available within the application. Crop improvement teams need to print tags, labels, envelopes, and reports throughout the season. Customized templates made by trained EBS users, Printout Manager tool, available to support printing activities in many core breeding programs, such as Experiment Manager, Planting Instructions Manager, and Harvest Manager. Wherever users see the printout icon, they can select the desired template and export the information that they need.
I will be presenting the highlights for the different capabilities developed by Breeding Analytics for Milestone 6. For statistical design model, the BA team enabled the uploading of design generated outside EBS. The team implemented the different validation rules for designs currently available in the system. Through experiment creation, user may now upload design for RCBD, augmented RCBD, and PREP design. Short description of the design can now be displayed in the UI. A new tool has been developed this milestone in BA called the Phenotypic Data Manager or PDM, which can be accessed via Experiment Manager. User need to choose an occurrence with trait data or with the status trait data collected. The PDM is an exploratory data analysis tool that will allow users to assess the raw phenotypic data before running the desired analysis. The tool provides the calculation of simple statistics, viewing of the uploaded raw phenotypic data, detect possible outliers, suppress data points. we will be enabling the integration of PDM with Analysis Request Manager or ARM. For ARM, user can choose one or more experiment for analysis, and user can request for multi-experiment analysis. For this milestone, user can see all the numeric traits collected for the chosen occurrence or occurrences. Note that in previous milestone, only common continuous trait will be displayed in this drop-down box, and only traits common across occurrences are listed in the previous milestone. Request name has been improved so that it will be more readable to the user.
One of the notable accomplishments of Milestone 6 has been work on the development of a complete genotyping workflow. In the service management domain, users can now initiate genotyping service requests. To do that, they can start by creating a service request by choosing a list of plots or packages that they have created in core breeding. They can then go on to provide basic information about the request and who is responsible for the request. And then they can go on to provide additional details about the service that they would like performed. Once they have submitted this, the lab is able to log into the Genotyping Service Manager and see all of the requests that have been submitted. Once they approve the requests, they can then use them to generate a genotyping batch composed of one or more requests. When they're setting up their batch, they are able to specify additional details about the technology that will be used and the markers that should be included in the assay. They then go on to generate their plate layout, to set up their controls, and to generate their sample IDs. Once they have done this, the forms that they need to send to the vendor should be ready to download from Batch Manager. Once the results are obtained from the vendor, they can be easily uploaded to the GIGWA database. These files are then available for lab members to download, and they also become available for the users who submitted the request in the Request Manager.